So we are 3 past. It's time to begin with this webinar. Hello again. Hello, everyone, and welcome to a new webinar by the Global Plan Council. Just a very brief introduction to our organization in case this is your first webinar with us. The GPC is a global coalition of plant science organizations, currently 24, with the mission to raise awareness about plant science, to build a sense of community among plant scientists, and to represent said community of scientists in the global policy arena. A little about the logistics today. There will be a Q&A session at the end of the talk. If you want to send your questions, please use the Q&A tool at the bottom of the Zoom toolbar. The chat, as you can see, is open and you can send comments there and say hi. But for questions, please use the Q&A tool. That will make my task easier during the Q&A. So today we welcome a very special speaker, Professor Danielle Way, currently a researcher at the Australian National University. She started her career at the University of Toronto and then moved to a postdoctoral position at Duke University in the US. She worked as a faculty position at the University of Western Ontario in Canada for 10 years, and finally landed at the Australian National University in 2022 for her current position as a professor. Danielle Way is an expert in plant response to climate change. She studies how rising temperatures and CO2 levels impacts plants, in particular their impact on photosynthesis, respiration, water fluxes, and growth. She is a highly cited researcher since 2020 and manages multiple grants, including one from the Australian Research Council, an NSERC Discovery Grant, and a John Templeton Foundation grant, among many others. So today she will tell us all about the climate change effects on forests and crops. So Professor Way, the floor is yours.
All right, thanks very much. First of all, thanks, Isabel, for the invitation to speak. I'm just going to pull up my slides and make sure that that's looking good. So, Isabel, is that a thumbs up?
Yes, it is.
Okay, wonderful. So, yeah, I just want to say, first of all, again, thank you very much for the invitation to speak. As Isabel mentioned, I'm currently here in Australia, although some of the work I'm going to talk to you about today is coming from my lab in Canada, which I left just over 2.5 years ago. Okay, so let's get this going. All right, so I'm sure everybody who's on this call is aware that CO2 concentrations are rising in the atmosphere, and they've already risen 50% since the Industrial Revolution. So back in the late 1950s, when we first started measuring, CO2 concentrations were sitting around 315 parts per million. And at this point, we're up at around 425 parts per million. And that increase in CO2 has direct effects on plants. It also affects the climate system because CO2 is a greenhouse gas that traps heat. And so we've actually already had approximately a 1 degree Celsius warming in global mean temperatures. And as we continue forward as a society, we have to decide what we're going to do to mitigate this effect. And what you're seeing here is a figure from the Intergovernmental Panel on Climate Change, showing different potential future climates for the Earth, where this right-hand figure showing a 4 degrees Celsius warming is really a business as usual scenario. So we often think of this as actually a worst case scenario, but really this is just what happens if we don't change our behavior. Under those circumstances, you can see that there are a lot of areas, especially at high latitudes, which is where Canada is, where actually those regions will warm by between 6 and 8 degrees Celsius by the end of this century. So those are enormous changes that are much larger than the degree of cooling that we had during the last ice age. Now, I started off my interest in this area mainly because we know very well that climate affects plants, and it's an important control on both where you find your species, so their distribution, but also how productive they are. And just as an example, this is a map of Canada here that's showing the climate suitability for aspen, which is a deciduous poplar species. You can see this dark green area shows the current core of the range of that species. Under a future climate scenario with that degree of warming, what you would actually expect is that most of the distribution for that species actually ends up in the Arctic Ocean. So there's really extreme changes in the far north, especially to these sorts of climate changes. But at 4 degrees warming globally, we're going to see change everywhere. So while we know that climate affects plants, I think what I also find really interesting is that plants affect the climate. So it's really a feedback system between the two. And so when we look at these sorts of figures coming from global models, those models have to incorporate the really large carbon fluxes that occur between vegetation and the atmosphere. And what I'm really talking about here, especially, are photosynthesis and respiration. And so just to show you what I'm talking about, here's a cartoon that we published a couple of years ago where we're just looking at the terrestrial carbon cycle for the globe. So a very simplified version here. And terrestrial plants every year absorb about 123 gigatons of carbon through photosynthesis. So that's the blue arrow. And respiration from plants returns about half of that every year back to the atmosphere. Heterotrophic respiration from microbes and animals releases about another 60 gigatons of carbon every year. And anthropogenic effects like fossil fuel burning and land use change are emitting about 10 gigatons of carbon every year. And if you just sum up those 4 fluxes, you can see where the accumulation of CO2 comes from, right? This is 7 gigatons of carbon accumulating in the atmosphere every year, causing that rise in CO2. However, if under a future warmer condition where it's drier, if plants are inhibited and photosynthesis is suppressed by even just around 2%, even if you assume that that suppression of metabolism also affects respiration a bit, without any other changes, you can see that the CO2 buildup in the atmosphere becomes much more rapid, and now you're looking at an exacerbation of climate change. So we really need to understand how these sorts of fluxes from plants are going to change under a future high CO2 warmer world if we want to understand how fast climate change is going to occur. So today I'm going to talk to you about 2 projects. The first one, which is going to take up 95% of the talk, is looking at trees and forests. And then I'm going to end with about 5 slides just talking about some recent work we're doing looking at wheat and climate resilience in crops. So the first topic, we'll look at northern forests in North America and their responses to rising CO2 and temperatures. And then we're going to be looking at wheat responses to the combination of rising CO2 and temperatures. So for those of you who aren't really studying boreal systems, just to give you a little bit of a view of where we are, this is the North Pole, and we're looking down at the globe, and you can see the boreal forest here circling the top of the globe in dark green. Now, this region is a really key player in that global carbon cycle, and that's because a third of the world's forest carbon stocks are found here, mostly in the peat moss and the soils in these regions. But they have relatively few tree species. So for example, across Canada back here, there's only about 14 tree species that make up that entire ecosystem. So it's not super biodiverse. And what that means for an experimental approach is that those dominant species have very large impacts on actually the global carbon cycle. And it's actually possible to study a large proportion of those species and figure out how they'll respond to climate change to improve our understanding of how the whole system might respond to rising CO2 and warming. So we undertook a really large integrated project to look at how climate change will affect the growth and the carbon fluxes in these northern forests. So it has multiple scales to it. I'm going to talk a little bit about each one in sequence. The first is we grew 5 common species of trees, looking at seedlings in a glasshouse, growing them under combined CO2 and warming conditions. To look at those responses in the field, we then looked at a large-scale experiment growing mature trees of some of those species in the field under elevated CO2 and warming. And then we also did a broad-scale impact looking at tree rings across the entire continent and across Canada to see how plants have already been responding to the historical changes we've seen in atmospheric CO2 and temperature. So I'm going to start off by talking about the glasshouse work. These are a couple of my students working in the glasshouse back in Canada. You can see these seedlings growing here for the deciduous species and the evergreen conifers here. And we had 6 treatments. So the trees were either grown at what was at the time current CO2 concentrations of around 400 ppm, or an elevated CO2 of almost double that. And then that was combined with either ambient temperatures that reflected where those trees actually come from, so the source of the provenance of the seed, and a +4 or a +8 degree warming. So this 8 degrees is what we're seeing under that business as usual type scenario for climate by the year 2100 in these high latitude forests, and +4 is sort of a moderate warming in these regions. So I mentioned we have 5 common species here. These are the common names of them. So there are spruce, which are Picea, jack pine, which is a Pinus species, tamarack, which is Larix, and paper birch, which is Betula. 3 of them are evergreen conifers. We have 1 deciduous conifer, tamarack. It loses its needles every winter, and then a deciduous broadleaf species. And I just want to do a shout out to the people who actually did all the measurements and the work. This paper was led by my student Julia Hammer, at the time dressed here in her Halloween outfit as a Ghostbuster using the gas exchange systems from ICOR. So just to orient you to the sorts of results you're going to see, I just want to emphasize that when you grow plants at higher temperatures, you usually get a shift in the temperature sensitivity of photosynthesis. So this is a conceptual figure that we've published showing measurement leaf temperatures versus the actual photosynthetic rate on the y-axis. And for a plant that's grown at cool temperatures, it usually has superior performance at these low temperatures compared to a plant that's grown in warm temperatures here in red. But the plant that's acclimated to these higher temperatures and has been grown at them usually has superior photosynthetic performance at the highest temperatures. So this is what we see in the vast majority of cases. And I'm going to show you, just to start with, a single species just to orient you to the measurements that we're going to be looking at. So this is for white spruce grown in one of the replicate years of the experiment. We measured across different leaf temperatures, and this is photosynthesis, so it's assimilation, measured actually at the growth CO2 for the plants grown at ambient CO2 and grown at elevated CO2. So if we just start with the ambient CO2, the plants grown at control conditions at their normal growth temperatures are in gray. The plants grown at +4 are in yellow. And the plants grown at that business as usual extreme warming of 8 degrees Celsius are in red. And what I think you can see really clearly is that we're not getting this sort of canonical, typical response where the plants are acclimating to the warming. Instead, what you're seeing is that every time you increase the growth temperature, photosynthesis is being suppressed. Now, if we look at the plants grown at elevated CO2, they're also measured at high CO2. So that stimulates their photosynthetic rate when you measure them. But again, you can see that we're not getting this typical acclimation response, and that as you increase growth temperature, you're suppressing photosynthesis. So I'm going to scale out, and I don't expect you to take in all of these graphs, but here we have the 5 different species. We did this experiment in 2019 and again in 2021. Obviously, in 2020, there was this minor thing called COVID that prevented us from replicating the experiment. But I just want to give you the overall impression here that the red line's always at the bottom for all of these species. And in general, the gray line is at the top, which is to say that the plant's photosynthesis is being suppressed in all these species as you increase warming. There are some standouts here. So in the deciduous species, such as tamarack, especially at high CO2, some degree of warming, that moderate +4, actually does stimulate the photosynthetic rate. But you're really not seeing that in these evergreen species. So if we think about how photosynthesis acclimates, there's usually 3 main processes that can be simplified down to, where a change in net photosynthesis that we measure is usually underlain by some change either in stomatal conductance, so the ability of CO2 to get inside the leaf to be fixed, photosynthetic capacity, so you'll see this term here, VC max. That's the maximum carboxylation capacity of the enzyme ribisco. So it's the ability of ribisco to fix CO2, and Jmax is the ability of electron transport to create the ATP and NADPH needed to fix CO2 in the Calvin cycle. So the capacity of this is what you're seeing inside the leaf. And then you also have CO2 losses from respiration. So changes in any of these 3 different processes could account for the shifts that we're seeing in the photosynthetic rates of these different plants. And so when we looked across these, the one that stood out really is photosynthetic capacity. So here again is that maximum capacity of ribisco to fix CO2. for the plants grown at ambient CO2 or elevated CO2. And again, we're just looking at the one species. And what you can see is that here's the plants grown at ambient CO2, ambient temperatures in gray, +4 in yellow, +8 in red. Every time you increase the growth temperature, the photosynthetic capacity, the ability to fix CO2 biochemically inside the leaf, is going down. Similarly, in the plants grown at elevated CO2, we see a very similar response. And so again, if we scale out and we just look broadly across all of the species across the different years we ran this experiment, again, you're generally seeing that the plants grown at the +8, the highest degree of warming, have a real suppression of this photosynthetic capacity, and the plants grown at the current temperatures usually have the highest performance. And again, some of these deciduous species like tamarack show you that that small degree of warming, the +4 warming, actually benefits the plants grown at high CO2. So we also noticed that that ability to maintain carbon uptake in the treatments correlated with the growth of the species. So here are just some pictures to give you examples where we have the plants grown at ambient CO2 on the left, those grown at elevated CO2 on the right. And within each of the species names, the arrows, the direction indicates current temperatures, +4 warming, +8 warming. And so in all of the species, +8 degrees warming really suppressed the growth of the species. And sometimes I think in the literature, this isn't expected because boreal forests are very cold, they're very temperature limited. And so one of the expectations is that warming might actually benefit these species. But here you're seeing that when they're well watered and they're given all the nutrients they want in a glasshouse, instead it's suppressing their growth. Now, as we might expect, growth under high CO2 is actually stimulated. And that's particularly the case for the deciduous species here, tamarack and paper birch. And those species actually, again, especially under high CO2, the +4 warming in the middle here actually grows relatively well. So I mentioned that photosynthesis acclimates to warming, and we've looked at this image before, but the other thing I want to show you is this shift in the thermal optimum that we usually expect to see in plants. So usually if you grow a plant at this higher temperature in red, you see an upward shift in the thermal optimum of photosynthesis. So we looked at that across all these species and across all the CO2 and temperature combinations. So that's the thermal optimum of photosynthesis here for the plants under either ambient or elevated CO2, and the different growth temperatures, 0, +4, +8. And we were really quite surprised to see that we do see acclimation to growth temperature, but only in 2 species. Is it significant? We see a response in paper birch here, a growth temperature effect, but it's really not anything that I would describe as what you would expect to see with these increases. And so I'd like to point out, especially to any of the students and postdocs who might be in the audience, we'd done a lot of studies previous to this where we'd only studied 2 species and we'd always seen thermal acclimation and this upward shift in the optimum temperature of photosynthesis. And I'd like to highlight that it was these 2 species we looked at. So, you know, when you do get reviewers pushing back on you about your ability to make generalizations, I'd just like to highlight that I am now a fan of that statement. Okay, so the results here are not guaranteed that you're going to see this type of response. So just wrapping up what I've told you for this first part of the study, warming suppresses photosynthesis, and the critical degree of warming that does that varied between the species, with the deciduous species being less vulnerable to it. The low photosynthetic rates we measure are really correlated with a reduction in photosynthetic capacity, so the biochemical ability to fix CO2. We're only seeing shifts in the thermal optimum of photosynthesis in some of these species, and the warming reduced growth, especially in the evergreens. So when we're talking about, you know, in my title, I wrote winners and losers of climate change, especially in this system, it really does look like the evergreen species that we think of as dominating these forests are really the ones that are most vulnerable to these changes. And instead, the deciduous species do seem to be able to tolerate that better and have greater plasticity for the higher CO2, higher temperature future conditions. So of course, what I've shown you right now are just seedlings grown in pots, and I'm telling you that that's going to affect the forest, and I can tell you something about how this forest will change. But to really be able to connect with what real trees are doing in the field, we're connecting our results with the work that we're doing at the spruce experiment. And so for those of you who've never heard of spruce, It's the Spruce and Peatland Responses Under Changing Environments study. This is funded by the US Department of Energy, and it's a large-scale experiment in Minnesota with open-top chambers that you can see here in a boreal forest system that has two of the species that we were looking at previously. So this is a close-up of what that looks like if you're inside one of these chambers. There are 10 chambers that have both below and above ground warming and either ambient or elevated CO2 conditions. The warming ranges from anything from no warming all the way up to a +9 degree warming, so very similar to what we were doing in the glasshouse. And the CO2s are also very similar. They're ambient or they're raised about double that, 850 ppm. And I'd just like to point out that those two species that I already mentioned were the ones that we do see the shift in the thermal optimum in the seedlings. Those are the two species dominating here. Black spruce is the green species that you're seeing in these pictures. And here in the autumn, you can see the tamarack starting to turn yellow because it's going to lose its needles. So again, just a shout out to the team that was doing the work. This field study was led by Jeff Warren, who's from the Oak Ridge National Lab in the United States. And two of my lab members, Bridget Murphy and Dr. Eric Dusenge, contributed significantly to those measurements with a lot of kit and a lot of gas exchange systems from LICOR. So really briefly, what I just want to show you is that when you go out and you look at these trees that are like 50 years old and that have been grown under high CO2 and warmer temperatures for a couple of years, you see this shift in the thermal optimum of photosynthesis, which is shown here on the y-axis versus the degree of warming that those plants are receiving. So both in black spruce here and in tamarack over here, you see that as you increase the growth temperature around the trees, you also increase the thermal optimum of photosynthesis, showing that photosynthesis is acclimating. And in tamarack, we also see an additional effect where the plants grown at high CO2 actually have a higher thermal optimum than the plants growing under current conditions. Now, what's interesting about being out in the field and working with these large trees is it's much more ecologically realistic. And we can also look at how the performance of the trees is related to the recent conditions they're experiencing in the field. And so this is a figure showing both of the species in tamarack and black spruce for the plants grown at different temperatures and either the ambient CO2 in white or the elevated CO2 plants in gray. And what you're looking at here is the difference between the temperature of the thermal optimum of photosynthesis and the actual average leaf temperatures that the plants are achieving in the field over the previous week or so. And so anything that's below zero simply means that the leaves are generally operating below the thermal optimum of photosynthesis. And anything above zero means that those plants are actually experiencing temperatures above the thermal optimum of photosynthesis. So although the thermal optimum of photosynthetic processes is shifting, it's going up as you increase the temperature of the trees, it's not keeping pace with the actual increase in the temperature. And so by the time you get to about a 4-degree warming in these trees, it doesn't matter if you're at ambient CO2 or elevated CO2, the leaves are operating above the thermal optimum. And that's problematic because if we look at just a sort of general figure here of looking at air or leaf temperature versus the photosynthetic rate that the plants are achieving, here you're seeing a cool-grown plant in blue, a warm-grown plant in red. As we're seeing in these trees, the thermal optimum shifts upwards. So that's what we would expect to see. But in this case, the thermal optimum is only moving by about 2.5 degrees, and the growth temperature is actually a 4 degrees difference. So the cool-grown plant is normally operating somewhere down here below the thermal optimum, where that variation in temperature on the x-axis doesn't really change the photosynthetic rate much when you look at the y-axis. But for the warm-grown plants that are operating up here as a starting point, even small increases in temperature are really going to cause a big suppression in the photosynthetic rate, which is why you normally see plants operating below their thermal optimum. So we're pushing them above that in these situations. So the question is, to what extent can we actually think about how forests are responding by using these large-scale experiments? And do we see evidence for reductions in growth or reductions in carbon uptake out in the field? And so this is work that was led by our postdoc, Dr. Achaïen Mirabel, working with Martin Girardin from the Canadian Forest Service and Juha Metsäranta, also from the Canadian Forest Service, and Peter Reich from the University of Minnesota. And so the question is really, do we see these detrimental effects that we might expect from the glasshouse and from this big spruce experiment in the real forests over the last few decades? So Achaïen spearheaded a new tree ring database for across the country with over 300,000 cores for trees from about 4,000 sites across Canada. And one of the first things that I think is really interesting that we get from her results is that if you look across the years where we can get data out of the rings, and you just look at the shift in the species that you find over time in these field sites, you can see that that's changed, especially in these 2000s. And the only deciduous species in this list is Populus tremuloides. So I mentioned earlier that based on the glasshouse work, it really does look like the deciduous species are better able to grow and tolerate warming and the rising CO2. And that is indeed what we're seeing if we go out and look at how prevalent these species are in the field. And instead, we're seeing drops in a lot of the other evergreen conifers that are in this list. So when we look at her data, what we did was we analyzed the growth sensitivity of all these different species to changes in temperature and also precipitation since about 1950. So the different species are listed here. The temperature sensitivity is shown here between yellow and red. Basically, anything in red just means that that species is very sensitive to temperature when it comes to growth. It doesn't tell you whether that's responding positively or negatively, just that it's sensitive. Similarly, for precipitation, anything in dark blue, that species is very sensitive in that region to changes in water availability. And what we found when we looked across the data was that in almost a third of the sites, or a bit more than a third of the sites, we find a negative growth response to warming in the trees, but it's actually the warming that happened last year. So if you're looking at a tree's growth this year, what actually matters for it is the climate it experienced last summer. And our hypothesis around this is really that in these trees that go completely dormant in the winter, they store up carbon this year, and then they store it to be able to grow next year, next spring after the winter. And so it's really the carbon storage that you accumulate in year one that allows you to grow well in year two. On top of this, more recently, we've been thinking about to what degree the growth suppressions that we're seeing in the field are really related to warming, or to what extent they might be related to changes in vapor pressure deficit. So for those of you who are not really used to thinking about vapor pressure deficit or VPD, you can think about this really as an atmospheric drought. Even if a plant has water in its roots and its availability, if the leaves are exposed to very dry conditions, then that closes the stomata and suppresses carbon uptake. And so across this region, you can see that there has been an increase overall in the vapor pressure deficit that this region has experienced since the 1950s by about 0.1 kilopascals. For those of you who do gas exchange like myself and measure photosynthesis, this seems like a really small change. So if I'm measuring a leaf inside a cuvette, I'm not even going to worry about that type of a difference normally. But when you look at the effect of this on the growth of the trees in this region, here are two of the most common species in the boreal forest, black spruce and white spruce. Here's how the VPD in those sites has changed over time. And here's how the growth of those species has changed over time. And this is the correlation between those. And so what we see is that for every about 0.1 kilopascal rise in this atmospheric dryness, the growth of these dominant species is actually declining by about 10%. So they're very sensitive to this change in atmospheric dryness. So coming back to the beginning of the talk where we talked about these sorts of models that predict what a future climate will look like, the question could be really, do these models actually capture these sorts of lagged effects I've just discussed, where dryness and warmness this year affects your ability to grow next year in these perennial species? And I guess the title of my slide tells you what we found. But here we have the data that I've just been discussing with you, where this is really an indication of the growth effect of these different climatic variables. So everything in blue is some measure of precipitation. Everything in the red colors is a measure of temperature, both for this year and for the previous summer and autumn. And basically anything that's negative means that as this temperature goes up, growth goes down. Whereas anything in the positive direction means that as you add more precipitation, you get more growth. And what I think you can see really clearly is that when we look at different models, these different terrestrial biosphere models, and feed them in the data that we have and ask them to make predictions of how climate will affect the growth of these trees, basically in these cold limited regions, they all assume that warming, all these reds, will enhance tree growth. And so they're not accounting for the negative impacts of having a warmer year on growth next year. And again, we think this is because they're not able to account for carbon storage and the difference between when carbon is stored and when it's used in these species that grow in this perennial way. Okay, so just to wrap up the first 90% of this talk, I'm a Canadian who has just moved to Australia. And so, yeah, as you can imagine, many Canadians and people who live in these cold regions would really like to escape the winter. And this is actually not a picture from Australia, it's a picture from Canada, where Canadians in the summer actually bring in a crane, install some palm trees on a beach on the Great Lakes in like these northern regions to try to pretend that they're in the tropics and that it's a nice warm region. Despite our trying to fool ourselves this way, these northern regions really are cold adapted areas where as you increase temperatures, it's going to have a serious impact on the forests that we have there that are so valuable, both for our economics and for their ecological value. So I just want to emphasize that overall what we found is that warming suppresses the carbon uptake and the photosynthetic capacity and the growth in the seedlings of these species. And we're seeing this across all of them, really. We are seeing some degree of acclimation, both in the seedlings and the trees, but it's really not keeping pace with the warming that we impose on them. And we are seeing negative effects of warming and also this atmospheric drought or high vapor pressure deficit on forest growth. We can see that in the tree ring data, but it's really not well captured by the current models that we have. And that's something we need to be able to work on. Oh, sorry, that skipped forward. Okay, so I just have like 4 or 5 slides total to just give you a little bit of a taste of some of what we're doing. Having moved to Australia, I'm no longer focusing on boreal forests, but before I left, I had actually already started doing some work on crops and looking at climate responses in crop species. So a big goal in our lab really is to identify climate resilient plants. So I told you how in boreal systems, it's really the deciduous species that seem to be able to do better, that they're sort of the winners under a climate change scenario. And so what we're doing is really characterizing plants under the future CO2, VPD, and temperature conditions that they will experience, and then seeing how they perform. And for crops, that really means not just looking at yield, but also looking at the nutritional quality of the seed that's produced. And that's because under high CO2, and we still don't really understand the mechanism, under different crops like wheat, rice, and maize, you see a decrease in the protein content in the seeds, a decrease in iron, zinc content, and other micronutrients that are all suppressed, making the seed less nutritious. So really briefly, we're doing a couple of different studies to look at whether or not there's variation in different wheat varieties for future climate resilience. And so this project started a few years ago in Canada, where we're looking at 20 different Canadian wheat lines. And that's in collaboration with Raju Sulanayakanahali, who's from Agriculture and Agri-Food Canada. So he's a government scientist there. And my current student, Zhizhong Zhuang, who is looking over last year and this year at 90 different wheat lines from Australia. And in both cases, we're growing them under current CO2, current temperatures, and also future conditions, trying to see how their yield and their quality will change. And so just to give you, again, a little bit of a taste, this is some of the first results we've gotten from the Canadian wheat project, where again, we have 20 different lines, and I'm not showing them here by line yet, grown either under ambient CO2, or a high CO2 of 750 ppm. You have grain yield per plant here. Plants grown current temperatures, a warming of +4, current temperatures, or a warming of +4. And in this case, the baseline temperature is the temperatures in Saskatchewan, which are the wheat growing prairie region of Canada. And so I think you can see that under warming, under ambient CO2, you get the strong decline in yield, although you see much less of an effect under high CO2. But if you compare how plants do under the current climate of current CO2 and current temperatures with future CO2 and future temperatures, we're actually getting approximately a 13% decline in yield on average. But we're really interested in the sorts of lines that are up here, which actually are able to benefit and make use of that extra CO2 and that warming to produce lots of seed. And so that's the sort of variation that we're trying to identify right now. So we can look down at traits and genes that are responsible for that climate resilience. Now, similarly, if we look at Australian wheat, this is work by Zhang Zhang. Here we have the plants grown under either current CO2 and their current temperature from the wheat growing regions of Australia, and a future condition of a +5 warming, which is predicted for the end of the century, and basically a doubling of CO2. And here again, this is across 30 different lines of wheat. And what we're seeing on average is about a 17% decline in yield. That said, again, there are some plants that seem to be able to perhaps better maintain their yield under future climates. And those are the plants that we're trying to identify and start drilling down into the physiology and the biochemistry to understand how they can do that. So watch this space is what I'd say. And I guess I just want to wrap up by pointing out that there have been over the last couple of years in various parts across the planet, this is from Canada, severe effects of high temperatures and heat waves on our ability to produce crops and on food security. And we really need to be looking at how those plants and how those different lines that we depend on perform under the future climate conditions that they're going to be exposed to. And so if we can find which plants and which traits and which genes confer resilience to higher temperatures and higher CO2, that'll give us a more secure base for our economy and for feeding people. And so very last point, I just want to say that this is actually one of the reasons I moved to Australia, is that not only can we do that sort of work here, but I'm also the director of the Australian Plant Phenomics Network here at ANU, where we have a lot of high throughput tools that we can use to actually really ramp up the number of plants that we can look at and try to identify which of them have those climate resilient traits. And I just want to highlight that the APPN is not just for people here at ANU, but it is actually open to international collaborators or people who have projects that they'd like to do. So if you're interested in talking to me about any of the work I'm doing, or just about exploring possibilities through the APPN, I've just thrown my email up here, and feel free to reach out and contact me. So with that, I just want to thank all the people who funded the work that I've shown you. I want to thank you all for listening. And with that, I'd be quite happy to take questions. I'll just stop sharing and we'll go from there.
Thank you, Danielle. It was a great talk. I had a question during the first part of the talk, but then questions from the audience have started coming in. So because I will have you probably at the end of the meeting just for myself, I will pose the question then.
No worries at all. That's great.
So let's go to the Q&A tool. Remember, all attendees, post your questions in the Q&A tool. I've seen a couple of questions in the chat, but I'm not going to be able to scroll the chat all the way to find it. So please post your questions in the Q&A tool. So we will go by order of the most the questions that came earlier plus votes. So I will go first with the question by Lucy Buki. For the greenhouse experiment, I think she meant this question came on 15 minutes after we started. So for the greenhouse experiment, when you apply +4 or 8 degrees treatment, do you increase the temperature homogeneously during night and days and all seasons, or did you use constant temperature anyway?
Yeah, so for the glasshouse work on the boreal species, we actually had programmed in the control temperature is actually the average daily temperature. So it goes up every day and then comes back down towards the end of the year. So it's following the growth temperature that you would expect to see at the site where those plants normally grow. We then add just +4, +8 on top of that. So under a climate change scenario, we do expect that nights will warm faster than days, but we weren't really looking at that effect in particular. So we just added +4, +8 to that baseline temperature. But the temperature itself was a realistic shift that started off very cool in the spring, sort of got warmer over the summer, and then came back down towards the autumn.
That sounds complicated to set up.
It's yeah, well, the system we could actually just upload a file, but here in the glass houses we're doing for the wheat, it would have to be input by hand. So instead we're doing sort of monthly average temperatures. So it's changing every month, but not every day.
Mhm.
Yeah.
Okay, perfect. So we will move to the next question by Sandra Kolichnik. As always, sorry for butchering all your names. So how long would plants need to acclimate to higher temperatures to see the reference conical curve? This came also 18 minutes after we started. Does that happen within one season or need longer time?
Yeah, so the ability of the photosynthesis of existing leaves to change in response to a change in temperature, usually you start seeing shifts. Like if you actually measure plants out in the field and you measure them every week or so, you'll find that the photosynthetic capacity and photosynthetic rates are shifting and responding to about a 7 to 10 day window of temperature. That's sort of what I've seen in the past. So it actually only takes a couple of weeks to see
Oh,
some sort of a shift. But if you develop a new leaf instead at the new growth temperatures, you actually are able to change the structure as well as the biochemistry inside the leaf, and that actually allows for a much stronger acclimation. So there can be constraints. An existing leaf doesn't acclimate quite as much as a leaf that grows under the new condition, but usually a couple of weeks is sufficient to see a change.
in fact, next question it's precisely about leaf structure and leaf area
Okay.
by Bruce Osborne. Can you tell us what happens to leaf area in response to increasing CO2 and temperature?
Yeah, so I think it's a question of whether this is a general question or not. Like in a lot of plants that are grown at high CO2, they have extra carbon availability, and that can increase like the canopy size if they're thinking about like total leaf area as opposed to the size of an individual leaf. Um, the leaves also become thicker because there's more carbon to build structure with, so they tend to become thicker and denser under high CO2. You often actually get the opposite under warming. So the plants tend to be thinner and a little bit more delicate. And so the two of them tend to offset each other to some extent.
So, in average, they stay more or less the same, or it depends on which one of these stresses pulls harder?
Yeah, if you have like a big jump in CO2 and only a little bit of warming, you're going to see mostly the changes that you would expect in high CO2. So the leaves will be thicker and perhaps bigger. But under a strong degree of warming with just a little bit of extra CO2, I would expect to see sort of these thinner, more delicate leaves that you often get. Yeah.
Okay. Interesting. So then we go with the first question that we got from Edwin Garzon. So sorry.
That's okay.
What role should neglected and underutilized crops play in building resilience in food systems at a global scale? This came 5 minutes after we started.
I mean, I think it's an excellent question. I do think that even when you're looking at the really big crops that tend to be grown in large quantities across the globe, there's still a lot of specialization within those crops that, I'm looking at Australian lines versus, Canadian lines. There's a lot of specialization there, but we are really vulnerable, I think, to the fact that we depend on such a small number of species, right, to feed so many people. So I guess in my own opinion, I would say that being able to increase, grow more of the crops that are already actually performing well under these higher temperatures, for example, and drought stressed, being able to diversify the food system I think is just going to add resilience to it. So it's not something I particularly study, but just in a general sense, I would say that's certainly my take on it, that anything we can do to build in resilience, given the scale of change we're expecting, would be beneficial.
Yeah, I was impressed. It was not related to crops. You were talking about the biodiversity in the Arctic forest, that there more the major percentage of this biodiversity is just 13, 13 kinds of trees. I was very impressed and kind of a bit scared.
Well, it's one of the reasons that it's kind of a more tractable system to study, right?
Yeah.
If I wanted to do this sort of work and look at the tropical forests, you have so many species, you're trying to find a way to actually simplify. Are you looking at fast-growing species or, early developing species versus late successional species? But in this case, we actually originally were growing 9 different plants from the boreal species, but some of them just don't germinate or grow very fast, and we weren't able to get them to grow well enough to be able to take measurements. But the idea really was to try to get as many of them as we could.
Yeah, in fact, that was my question. Why did you decide to go for those 5 and why only 5? So more or
Yeah.
less you already answered.
Yes, yeah, yeah. So it would have been nice to have all of them, but some of them germinated and never got any bigger than this after the end of the first year. So yeah,
Yeah, that I think that happens a lot with trees.
they can be tricky.
Yeah, they
Yeah.
can be very tricky. So let me see. So I think I'm going to choose the question by Omatola Avidayo Olunuga. Sorry about again butchering names. Can precision agriculture, that means smart irrigation, shade nets, or breeding for thermotolerance, using CRISPR-Cas or any other gene editing technique, mitigate yield losses under chronic heat stress, in your opinion?
I do think that anything that we can do that will actually reduce the temperatures the plants are experiencing would be helpful, right? And so if you think about this from a general, the overall energy balance of a leaf or of a tissue in a plant, you know, you can shade the leaves, right? That does reduce the amount of light that they get. So
Mhm.
if it's during hot events, though, that's still going to benefit them compared to overheating with all the extra light. Um, Obviously, irrigation is a key tool for keeping the plants cooler. Um, but if we're looking at modifications for heat tolerance, I think we often think about things like heat shock proteins and these sorts of changes. But you can also think about how if you go out into really hot, dry regions, how do plants do this themselves? And a lot of the time, it means that they'll have, for example, hairs on the leaves, for example, trichomes that will reflect the
Mm.
light itself. They will have different colors. There'll be a lighter color. So I think there's a lot of different ways that we could play with how a plant looks that would actually give them an increase in heat tolerance. But it's something that really hasn't been done very much. We've talked about it here at ANU a bit about some projects that we could take on to try to actually start developing some of those tools and those plants and look at how that affects their yield. But we haven't gotten to it yet. So yeah.
Very interesting about the color. Uh, that caught my that caught my attention, curiosity.
Yeah, there's a lot of structural changes, right? Like if it's really hot and dry, if you look at a cornfield, the leaves will be straight upright and they'll roll to basically reduce their exposure to the light, right? That's shining at noon right above them really hot. They don't sit like this and absorb the light. They do this.
Mhm.
And so again, I think there's a lot of things that plants are inherently doing that we could perhaps co-opt in when we're thinking about how to make these more tolerant plants.
I will now remind the audience that to use the Q&A tool for sending the questions and for both your favorite ones that will make my task much easier in order to choose the questions to be asked. So I'm going again for a question by Lucy Buki. For the wheat experiment, what yield components are changing with temperature and CO2? Um, I think it's number of grains or size or of grains or tillering.
Yeah, so we have looked at all of these different traits. I don't want to give too much away since this is my PhD student's work and we haven't published it.
Okay.
No, but we are seeing changes mostly in it's usually correlated actually with the number of grains that are being produced. But
Number.
again, the different varieties are responding differently. Some of them increase tillering, some of them decrease tillering. We see an early stimulation of growth actually under the high CO2 and warming conditions. But by the end of the growing season, instead those plants have actually less biomass than the plants growing under the current conditions. So it's like they get an early start, but then as they start actually getting into the reproductive phase and into this sort of the middle of the summer, that heat load becomes too much and then it actually suppresses growth. So you're seeing shifts across the season as well in how this
Mhm.
is playing out for plants.
Okay. So, but the okay. I was going to make a follow-up question, but I think we are getting too many.
Okay, all right, that's fine.
Yeah, yeah. So next question by Elijah M. Getiro. By what factor will the 1.5, 4, or 8 degrees rise in temperature reduce crop yields globally due to reduced peace?
Oh, photosynthesis, that's
Oh,
photosynthesis.
photosynthesis, yes.
Yeah. Um,
In your opinion.
yeah. I think the link to photosynthesis is less well established, I think, in the sense that the warming is causing this reduction in photosynthesis, which then decreases yield. So there's a lot of things that could be decreasing yield. We're certainly seeing in our wheat work that it's actually seems to be so far the prime suspect across the things we've measured. Um, and I know that there's data out there. Dave Lobel has some data about looking at sort of average decreases in yield, I think, in wheat and other crops for every degree Celsius warming.
Mm.
And I honestly cannot remember the number right now, so I don't want to give the wrong one. But I do think it's around, sort of around the range of about 5% for every 1 degree Celsius warming globally. That's sort of the expectation, somewhere around there, but I'd have to look up the actual number.
Okay. So it's out there, but can you give the reference, or maybe not
Um,
right now? Maybe we
yeah.
can include it for later in the follow-up email that I'm going to
Yeah, if you
send to
do that,
everyone.
I can give the actual numbers and I can give some references for that. Happy to do that.
Mhm. Okay, perfect. In fact, I'm checking just I'm seeing a comment in the chat. Just as it come across my eyes right now. Adeline Mujiva, again, as always, she's asking for your email address. She would like to contact you. You have shared it already in the
Yes.
slides. So we
So
will,
I'm actually just going to put it in the chat myself right now
yes,
um, there we go.
yeah. So if you want or need to contact Danielle, that's her email address. So next question. I think we are going to go with Gary Bradbury. For many cereal crops, we need to think about the size and vigor of root systems. Yield and grain quality often affected by water stress during flowering and grain fill periods. So stress we can see is increasing. We need plant breeders to work towards more vigorous root systems, even though it is difficult. Do you do you agree with this and the audience? So that is the question.
Yes. Yeah. So I think, anything in the below ground sphere is usually understudied because it is difficult, right? So it's very challenging. In the sort of studies we do, we get around this to some extent by mostly providing ample water and fertilizer. And in that sense, what the data I'm showing you are really best case scenarios. These plants don't experience, extreme stress in any way except for slightly warmer conditions, right?
Mm-hmm.
Even the highest temperatures they experience in the air temperatures are around 34 degrees C. Like these aren't 40-degree stresses. This is sort of moderate. You wouldn't even expect it to be stressful per se. But the ability of those root systems to be able to get the water to help cool the plants and to grow them and to extract that nutrient is really yeah, it is understudied.
Mm-hmm.
And I know that there are drives to actually increase our ability to be able to do that. So here at ANU, I mentioned the plant phenomics network that we have. We're actually developing a whole root phenotyping pipeline. So these are rhizoboxes. So some of them are over 1 meter tall,
Mm-hmm.
where you grow species like wheat or maize and actually watch the root system develop, and look for differences in the root traits. So this is something that my student might be doing with some of the more promising lines for the wheat. We'll be growing them in pots, but we might be able to do some add-on experiments where we look for root traits. But this is definitely something that I think needs more attention.
Mm-hmm.
But it really does require the infrastructure and the tools to be able to do it. And I think that's been a big limitation.
Mm. Yeah, I would love to see those facilities, to be honest.
Yeah.
I'm always very amazed when I see videos of how it might work. So, in fact, we have another question that is also related, not directly with roots, but with soil and whatever happens in the soil. So, by Luisa Oliveira, was soil and soil health, particularly soil microbiology, considered in your experiments?
Yeah, so we weren't looking at the soils per se. So for the boreal species, we were growing them in sort of a peat mix, which is they often grow in peat bogs or in areas that have a lot of peat. But really, we're just looking to make sure that they have the nutrition that they need. So there's no nutrient stress and there's no water stress. But we weren't looking at the soil microbiomes at all, or the impact of mycorrhizae, for example, on any of these traits. I've had other students work on mycorrhizal studies actually, under high CO2 and temperatures, but we've seen sort of mixed results. You might often expect that the plants that have mycorrhizae would be more tolerant to stress under higher temperatures, but that's not always the case, at least in our studies.
Mm-hmm.
And one of the hypotheses is that the extra carbon sink that is needed to support those mycorrhizal species in a warming situation where perhaps photosynthesis is limiting and carbon is limiting growth, that actually is what tips them over into mortality in some of the studies that we're doing. But that's work that we haven't published yet. So yeah.
Mm. Again, in the chat, I see a question that I want to, well, it's more like a comment by Ridley Opasa. Uh, they say, I'm curious for a replica of the study also in the tropics. Yeah, I think they refer to the results in the forestry that you presented at the
Yeah.
beginning.
Yeah, so there is a face experiment being built in Brazil. So there is that sort of a system being built there, although it doesn't include the warming. Um, and I do know that there's a fair bit of research that's being done at STRI, so the Smithsonian Tropical Research Institute in Panama, where they
Mm-hmm.
look at some of these species. Um, we have done some work. So the first author on a lot of that work was my PhD student, Eric Dusenge, who is now actually here with me in Australia, not in my lab, but actually in a separate lab. And he is originally from Rwanda and has done some field studies there with colleagues in Sweden, where they're looking at warming experiments on tropical montane species.
Mm-hmm.
And similarly, what we're seeing is sort of differences in the ability to acclimate. Photosynthesis and respiration seem to correlate a lot to how well those trees grow under the warming conditions. But yeah, warming by CO2, it just requires a lot of infrastructure. It's one of the reasons I'm here at the ANU, and my previous institution had really good facilities for it. So doing CO2 in a glass house or in a growth chamber is usually not too challenging, but the expense of running these big CO2 and warming experiments in the field it takes years to build them and then a lot of money to support them. So that one that you saw in the boreal forest, that was, like I said, that was a US government funded project. So it took them quite a long time to engineer and get it right.
Mm. Yeah, that's true. That for some very important research, I would say, because this is important research because we need it for the future in order to prepare and understand and then prepare. You're right that the infrastructure is a very limiting factor, and that makes it only possible for some researchers in some regions to really undergo these studies.
Yeah.
So we have a next question by Mohammed Hamad. It's a long question, so
Okay.
be,
Yeah.
be prepared. Um, Given that rising temperatures and CO2 levels can initially enhance photosynthesis in some forests, but over time lead to negative impacts like thermal stress, drought vulnerability, and altered nutrient cycling, how do we quantitatively disentangle these competing effects to predict long-term forest carbon sink stability? Let's start with that part.
Um, so I think, there is obviously a lot of evidence for the carbon fertilization effect. So that's just the ability that as you have rising CO2, there's more substrate for photosynthesis. And so
Hmm.
have more carbon, they grow better. You don't see that in all places in the world. So we get our best evidence for this, both from tree rings, but also from these FACE experiments, which are the free air CO2 enrichment experiments. Um, and so, for example, in places like here in Australia, where the soils are really low in phosphorus,
Hmm.
or any where you have really low nutrient soils, low nitrogen as well, basically the extra CO2 doesn't cause any real effect on growth
Oh,
because it's not carbon that's limiting the growth of the trees, it's the nutrients.
hmm.
Um, so
Makes sense.
that's yeah, so that CO2 effect you see most strongly in the areas which have rich soils and crops where you can fertilize them,
Hmm.
or where the nitrogen fixing like soybean. Um, so in forests, I think the thing is you do see this rise in growth and productivity under high CO2. Just like with temperature, though, photosynthesis acclimates to CO2 over weeks to months.
Hmm.
And so if you grow a plant at high CO2, the plants have enough carbon that they basically reduce the amount of photosynthetic protein they make, especially rubisco.
Hmm.
Um, they have more of it than they need. And so this actually tends to reduce the carbon fertilization effect that we might expect. So there's sort of these competing effects going on over time where over years, often what you can see is that if there aren't enough nutrients in the forest system to sustain that growth under high CO2, the plant growth declines over time. And we see that in a number of the forest face experiments across North America and Europe. Um, so in my guess, I think the main thing is that the temperature stresses, like high CO2 will benefit growth during certain periods and in certain places,
Hmm.
mostly, and more in some species than others. But temperature stress affects everybody.
Okay.
Um, so I do think that when you look at these extreme heat waves and their ability to basically kill plants compared to the stimulation of growth that you'll get year on year from high CO2, that these temperature effects and also, of course, the fires that come with them are actually going to be the really dominant players in determining where our forests go. That's my own personal take, but in a lot of the work that we do where we combine CO2 and temperature, the temperature effect is really dominant,
Hmm.
even in well-watered, well-fertilized plants. So I expect the same in real forests.
I think it's really important what you just said, that is there is no net positive for having more CO2 in the atmosphere, but there is a very net negative related to increased temperature.
Yeah, and I do think, that there is a net positive to the high CO2 direct effect. It's just not going to outweigh the negative effects of the temperature from
Hmm.
what I've seen.
Okay. And the second part of the question by Mohammed Hamad, are there emerging ecological and biochemical thresholds beyond which forest resilience collapses, and how can remote sensing or mechanistic modeling improve early warning signals for such regime shifts?
Yeah, so I'm not aware of sort of where those tipping points would be from a biogeochemical sort of standpoint or such. I do think that if you're looking at those regime shifts, for areas like the boreal forest where the deciduous species seem to actually perform better than the evergreen conifers, I do think you could pick that up through things like the remote sensing because, of course, in the winter the deciduous species are losing their leaves, and the evergreens are actually still there and they're dark green. And so you can probably start looking at things like canopy cover during the winter as well as just changes in the overall greenness, because again, there's just differences in how dark green the needles are compared to that sort of brighter green that you get in the deciduous broadleaf species. And I really haven't seen this in the sense of having seen studies about big changes in those species across the forests. But I would like to point out that it's actually one of the few negative feedbacks I've seen into climate change, where we talk a lot about all these sort of really problematic positive feedbacks, where, for example, if you have warming and it releases more methane from peat moss and soils in these forests, that causes more warming, which causes more release of CO2 from methane. And so you get a positive feedback. But in this case, if you get a shift in the species where these evergreen conifers actually their growth declines or their abundance declines, and instead you get these deciduous species, the conifers keep their needles year round and they're very dark. And so they actually absorb more radiation from the sun, and it leads to warming at these high latitudes because they're not reflecting the light. Whereas these deciduous species, even in the summer when they have leaves, they reflect more of the light back out to the back to outer space through the atmosphere. And in the winter, they're not there, so you get snow cover, which is white. So that shift in the albedo, that reflectivity of the entire ecosystem, would actually result in some net cooling at these high latitudes. So it's one of the few feedbacks I've seen that actually, instead of making climate change worse, would actually mitigate it to some extent.
Interesting.
That was a long answer, but
So I think we have 10 questions to go.
okay.
Yes, by Giggs Kleta. To what extent could changes in solar irradiation affect the impact of CO2 and temperature on photosynthesis?
I'm not quite sure if they're asking, if in full sun, or if they're thinking about, shading the plants versus what happens to the solar cycle. And I'm not quite sure.
I imagine they refer to the shading because we cannot control the sun right now.
Yeah. Well, that's why I was wondering what the question. So if you're thinking more about shading, I mean, we've always grown our plants under sort of lots of light when looking at this. It's a good question. I mean, under shading, you would expect that those plants might experience certainly lower leaf temperatures, so the warming may not be quite as detrimental to them. The high CO2, it depends if the plant is more light limited, then the high CO2 is going to provide perhaps a little bit less of a boost to the plants, because if carbon's not really your main limiting factor, if you don't have enough light energy to really fuel the uptake, I imagine that you might get less of a boost from that. But it's a really good question. I haven't really seen much work on how plants that are sort of maybe sun plants versus shade plants or leaves are actually responding to those different conditions.
Yeah, it's an interesting new factor to add.
Yeah.
So we have a next question by Arthur Nosalevich. Hope that's your name. It's a long question again. What about heat waves? Their probability is expected to increase. They are not so clear in terms of average temperatures, but they do not allow for acclimatation, for the acclimatation that you are talking about. We've made some experiments and they strongly decrease photosynthesis through their impact on soil water availability via increased evaporation, transpiration, and LBPD. Moreover, depending on timing, their effect on crops can be even stronger, as we all know. What could be the effect of increased CO2?
Let me tackle the heat wave part itself first. The way I tend to think about the research that I'm doing is really, again, this is a best case scenario. These plants, they're experiencing, 4 degrees warmer every day. So maybe they're going from a growth temperature of 25 to 29. Again, you don't expect that to be anything that's going to super stress or kill plants in any way. And they're getting lots of water and they're getting lots of fertilizer, and they're not experiencing things like heat stress or pest or disease. I absolutely agree that the heat waves that you add on top of this sort of a growth decline or a reduction in CO2 uptake are seriously detrimental. So to me, these are additive things that you have to layer on top of each other when you're thinking about where the system is going. So it's absolutely true. They're not going to lead to acclimation. The heat waves can seriously damage canopies, cause serious disruptions to the hydraulic function in the trees, and just cause mortality, right? So we see an increase in mortality, for example, in the forest work that we've done, even those seedlings that are the best case scenario. In the +8 warming, we get over 60% mortality in some of the species.
Wow, 60.
So, you add a heat wave on top of that and they're just going to be toast. So I think, again, you've got to think about this in an additive way. And then with crops, trees at least have multiple years where maybe there's a heat wave this year and they get knocked back, but they can continue to grow next year. With crops, obviously, it's really an issue with the timing. Get a heat wave during the period for reproduction, or especially things like pollen tube elongation, you just won't get any yield at all, right? So, and things like CO2 aren't going to really affect the ability to tolerate that heat during those key points. One thing I want to note, though, is that I think a lot of the time people think about, high CO2 will be beneficial, warming will be a problem, and they can counteract each other a bit. When it comes to temperature, though, high CO2 inherently increases leaf temperature. So if you don't change the temperature of the plant and you give it high CO2, the stomata close. And if the stomata are starting to close, they're transpiring less, they're cooling less through evaporation. And so if you look at face experiments, you can actually take a thermal camera and go out there. And you can see which are the high CO2 plots and which are the ambient plots, even in well-watered plants like crops, just by looking at the leaf temperature. So high CO2 can actually make heat stress worse because the plants are going to operate at an even lower stomatal conductance and an even higher leaf temperature.
Okay. So next question by Good Vutunu. Thank you so much, Professor Way, for the presentation. I kindly inquire if there is a big variation, or would you expect a big variation between Canadian findings in Africa, given the same forest and crop cover? So it's not precisely the same forest.
Yeah. As I've said, we have had people who've worked on some of these, at least the tropical montane forests in Africa. I think the main difference is that there's an expectation that in high latitude forests and temperate forests, that because they experience big shifts in temperature across every day and across seasons in the year, that they're actually able to cope with a wider range of temperatures. Whereas in tropical regions, there's sort of this hypothesis that because they tend to experience very similar temperatures every day between day and night, and also across the season, they don't go from -20 degrees Celsius to +35, that they may have a lower degree of plasticity to be able to acclimate just to those conditions. We're really lacking in tropical data. There really aren't enough studies. But there is some evidence, there is some plasticity in the ability to acclimate photosynthesis, but my impression is that it is somewhat lower than the ability of some of these other species in other regions. That's probably about as much as I can say on that, though.
I think next question is a good complements well the previous one and what you are saying. So Okon asks, in Africa, we have these equipment issues that hinder research like this, as we discussed. Is there a body that you can think of we could partner with for supplier funding for these small handle CO2 equipments to ease these researchers?
Yeah, no, it's a really good question. I know that here through the Plant Phenomics Network, we often do get requests from other countries where sometimes there are programs, for example, between Australia and another country to help promote agricultural stability or other research in those areas, where there's sort of funding through those government bodies to help support us to help develop the research capacity in an area or provide at least training and such on these. Um, Outside of that, I think right now it's a bit of a challenging environment globally for some of those sorts of entities.
Especially,
But
yes.
yeah, but it's and I'm just simply not aware of large scale funding opportunities for getting that sort of equipment. Um, Even for researchers in developed areas, these are really expensive and they're really difficult to access. Um, but like I said, for at least some of these, being able to take advantage of these sorts of institutions like the Plant Phenomics Network, where people can actually come and work here. And we actually have graduate student scholarships, for example, for students to be able to apply to do projects here. So there are some of those sorts of opportunities, but on a broad scale, I'm unfortunately not aware of them. And I agree, it's a big problem, right?
Hmm.
We're very fortunate at places like this to have this equipment. But I recognize that it's really not available to everybody.
Yeah, this is precisely why. So this is the reason why we don't have data about tropical forests.
Absolutely. This is absolutely what it is. Yeah.
So maybe they can contact you in order to be able to go to your institution. So I'm
Yeah,
going
I'd
to
be
put,
happy. Again, I put my email in the chat. I'm happy to chat with people about opportunities that I know of here or to talk about potential sources for that sort of support.
I'm posting Danielle's email address again, just in case it gets lost because it's very active, the chat.
Okay.
So we have a question by Iram Sharif. Please tell which traits could be used to screen crop plants against high temperature and give clear-cut difference between susceptible and resistant varieties.
Yeah, so actually with the crop work, we're really at the beginning of our own work on this. So I'm afraid I can't really give anything on that sense. I will say that our preliminary work here in Australia is showing that the plants that lose the most yield are the plants that are the least plastic or the least
Mhm.
acclimating when it comes to photosynthesis. And I just want to point out that I realize I have a background in photosynthetic physiology, but I actually wasn't expecting that to pop out because I recognize that things we've discussed, like roots and other sorts of traits, could be more important. But at least from from our preliminary data, we are seeing that it's actually the ability to match your photosynthetic processes to the environment you're in. So that ability to acclimate and be plastic, that seems to actually give some of the plants better yield under the future climate treatments.
So plasticity would be the key feature here.
But again, this is sort of like from 1 year of data. So I'd say watch this space because we're repeating things this year and we'll be working on that. It's
So
basically
next,
our question.
so next question is by someone, so they didn't leave their name. What is the relationship between BPD and temperature to yield?
So I think it's sort of complicated because at least out in the field, when you have warmer conditions, you almost always have an increase in the BPD. So these things tend to go hand in hand. You can separate them experimentally in experiments, but even for people with really good facilities, it's nearly impossible to create a warming experiment that doesn't also increase the BPD of the plants. And in some of our experiments, we do try to mitigate that. But at this point, I've simply decided I'm going for ecological realism.
Mm-hmm.
And so I don't worry about trying to mitigate the effect of the VPD by ramping up the humidity in the warming condition. I simply try to keep a realistic relative humidity for future climate predictions. So I really think that it's a combination of both. But that said, there's a lot of work that we attribute plant responses to temperature that I think might actually be more related to VPD. And so
Interesting.
we actually had some difficulty. We published a paper just a couple of years ago in Global Change Biology with a colleague, Walid Sadok, where we did a meta-analysis looking at just VPD effects on plants. And so these are all plants that are grown under different levels of atmospheric drought, right? So different drying, but they all have ample water. Some of them are even grown in hydroponics. So the roots have water, but the air is drier. And we found reductions in yield, reductions in growth, increases in total evaporative losses from the canopies, smaller leaf area, increases in things like ABA concentrations. So basically the plants are being droughted. They're just being droughted by the air. And so when you're doing a warming study and you think that everything you're doing is related to the increase in temperature, I would just challenge people to think a little bit about to what extent maybe it's the VPD that's causing the responses they're seeing, and to what extent it's the temperature. And I think the importance for that is actually that when we think about these big scale climate models we use, if we want to predict how plants will grow in the future, we need to be able to predict there are some places in the world where it'll get hotter, but it'll still stay relatively wet. And there are places where it's going to get hotter and much drier. And the plants could respond really differently to that change.
Yeah, to two scenarios. Yeah.
Yeah.
So we have a next question by Violeta Salazar. Considering that increasing CO2 increases photosynthesis at high temperatures, then it could be considered that controlling stomatal closure is essential under these conditions?
Yeah, so higher CO2, again, you normally expect to see that if you supply the leaf with high CO2, that they will have higher photosynthetic rates at these high temperatures because the high CO2 is suppressing photorespiration. So we definitely see that. And I think, if you think back to where I sort of showed one of the first slides with data where we measured the photosynthetic rates under high CO2 and they were much higher in the glasshouse work, we do see that sort of an effect. At least in the glasshouse work that I showed there for the boreal species, there really was almost no acclimation of the stomatal conductance across these different species. So I was a bit surprised. And again, it could just be that in these species, things like spruce and pine have very low stomatal conductance. This isn't something like a C4 plant or like wheat that has really high rates. But yeah, so we weren't really seeing big stomatal differences there. And in the wheat work that we're doing here with the crops, we will be looking at changes in stomatal anatomy and conductance, but we don't have that data yet.
Thank you. We have 2 questions left. So a question by Madam Palsine. Dear Way, it is an excellent presentation. Just I wish to know about the changes in soil enzymes, including soil microbial biomass under elevated temperature alone, elevated CO2 alone, and a combination of both elevated temperature and CO2. Do you have
Yeah, so
data about that?
I can point you to something. It's not from our lab, but that's the experiment in the field, the spruce experiment with the big open top chambers. I mentioned when I described it that they have both above ground warming and also below ground warming. So in those plots, each of the plots is actually hydraulically isolated from the soil around it. They've driven down like sheets of metal into the soil. So there's a plot below ground as well as above ground that you're seeing. And then they've driven down rods that actually warm the soil between 0 and 9 degrees Celsius. So there's the full spectrum of warming and CO2 and its effects on soils. And from the point of view of the people designing the experiment, they were really interested actually in how those soil systems are going to respond to CO2 and temperature, because their main question was, for these big models, is all that carbon locked up in the soil going to come out as CO2, which is less potent as a greenhouse gas, or as methane? And so there's actually quite a lot of work, and a lot of that's now been published from that spruce experiment. So I would just encourage the person to take a look at the spruce. They have a website, so the spruce at the Oak Ridge National Lab, or if you just type in the word spruce, CO2 and warming in your search engine, you'll find that, or just look for the papers. But they have a lot of information about how it's changed the microbial communities, the carbon stores in the soil, the gas fluxes. So there's actually quite a lot of work on the below ground that came out of that experiment.
Great, perfect. Next question is by Alan Measures, and this might be the last question. Has the world's capacity to produce biomass already peaked? If not already, when might it peak, in your opinion? and they add, I ask because of proposals to use more biofuels, that means aviation fuel and biomass to replace fossil fuel, plus the issue in higher income countries of our dietary reliance on animal agriculture.
Yeah, I think that when it comes to the issues in biomass, it's really about land use decisions, and also, to be honest, it's going to be about extreme climate events like fires. That's my guess. I do think that, you know, the warming temperatures, you do expect to see expansions of things perhaps like grasslands, but then again, high CO2, there's some evidence that it actually supports the invasion of woody species into grasslands in areas because plants have more carbon and trees require more carbon to be able to establish in those areas. Have we already peaked peak biomass? Again, I think that's going to be a bit of a fight, so to speak, between, you know, deforestation and things like fires and floods that are, you know, killing forests. And we are seeing an increase in forest mortality and tree mortality globally. There's a lot of work on that. But at the same time, there's a lot of interest in reforesting, and I think it really depends on the areas you're talking about.
Yes.
So if you're talking about, for example, areas in, you know, the eastern United States, areas that were originally cleared for agriculture have been allowed to grow back up as forests, and there's much more biomass there than there used to be. But of course, globally, we are cutting down a lot of forests and converting them into fields for creating food and also just places for humans to live. So yeah, that's a really thought-provoking question. But my guess is that overall, we're not going to see, in general, higher biomass in the future unless we decide to manage our landscapes effectively to do that as a way to improve carbon sequestration and ecosystem services. So I think it could be done, but I think it requires a lot of initiative and support from governments around the world.
Yeah, probably policy as well.
Yeah, absolutely.
Yeah, I think we managed to finish more or less on time, as we have set ourselves between Danielle and I a limit at 90 minutes, and that we were just reached that limit. So thank you everyone who has attended and has remained in the space until now. And we just to let you know what are the next steps. So I will prepare the video for uploading into YouTube in the coming days. Before 1 week, you will receive the link to the video in the channel, in the YouTube channel that I'm going to just share, just in case you are not following us there. And I will try to include in this follow-up email some information about how to reach Professor Way and how to connect with her for future communications if you wish to. And we are preparing a next webinar for May, but we don't have all the details yet. But I will, if I finalize the details next week, I will also include this information in your follow-up. And as always, watch this space, the Global Plant Council website primarily, but also our social media channels. You know, we are we are very active there. So Danielle, thanks very much. Has been a great talk. And you were great answering every question through your way. So I wish
And
we can.
I'll just add to anybody who's still there, if you do email or you have questions, it is Friday night here, so I will probably not be addressing them on the weekend, but I will do my best to get back to things next week.
Yeah, I can add that too. I will not upload the video on YouTube during during the weekend, so do not expect anything until at least Monday, Tuesday. So maybe we can repeat this in the future. Thank you. I'm not addressing you, Daniel, about that. And thank you, everyone, and see you soon.
All right. Thank you very much. Thanks.
Bye. Have a great Friday evening.
Thank you. You guys all have a great Friday day or evening. All right. Take care.
Bye. No, people are leaving the room.
Yeah.
And
Yeah, I wasn't sure if you wanted me to stay on at all to chat about anything before I go.
I'm ending the stream also on YouTube. I think it was great. You